the autonomic neuroeffector junction. So we've talked a lot about the neurons involved, the receptors, we talked about the neurotransmitter. Let's talk about the junction. Let's talk about the synaptic area of communication and see how these players fit into that communication. So we want to start by establishing some of the differences between our neuroeffector junction and a traditional neuron to neuron synapse, okay? So this is specifically the junction where we have that postganglionic neuron synapsing onto our effector tissue, our effector organ, heart muscle, cardiac muscle, glandular tissue, adipose, right? And so the synapse here looks different than the synapse at our postganglionic and preganglionic neuron, the synapse at our autonomic ganglia. So if we could just jump back here briefly. Let's look at this, right? This synapse looks very different structurally than this synapse. Okay, we'll talk about some of those differences, but this is the region that we're speaking about when we talk about the neuroeffector junction, whereas this is a neuron to neuron synapse, right? And so we want to establish that there are certain structural differences between those two regions. So the neuroeffector junction is larger, right? It's going to be larger and it's different in the width of that difference. So the area here is larger, the synaptic cleft, if you will. And then we also see the presence of what is called varicosities. So whereas at our neuron to neuron synapse, we've got our vesicles stored in the axon terminal, here we have our vesicles stored in this swelling that's called a varicosity, okay? And those are the two main structural differences between a neuron to neuron synapse and what we see here at the neuroeffector junction between the postganglionic neuron and the effector tissue. So I'll say that again, the two main differences being the larger difference, so it's a farther width for those neurotransmitters to diffuse across, as well as the presence of varicosity. So there's no axon terminal the way that we've seen in our neuron to neuron synapse. Instead, the neurotransmitter is stored in the varicosities, which are these swellings of the end of the neuron. Okay, so let us start by illustrating that. And we want to bear in mind here that we have to think about both divisions of the autonomic nervous system. Okay, so there's not a one-fits-all neuroeffector or a one-size-fits-all neuroeffector, route of communication. There are different players involved in the sympathetic side than the players that are involved in the parasympathetic side. So we're actually going to draw this out, illustrating both of those scenarios. Okay, oops. I apologize, this Zoom tab is getting in the way here. Oops, oh goodness. Okay, so we're looking at the neuroeffector junction. And we're going to divide our page here into two so we can think about both divisions of the autonomic nervous system. So we'll look at the sympathetic here. And we'll look at the parasympathetic here. I apologize, my husband's on a call and you may hear him. He's being very loud in the back there. Parasympathetic here on the right, okay? Let's first establish again those two main structural differences that we just mentioned. So we've got our postganglionic neuron, right? That is ending not in a discrete axon terminal, the way that we see in neuron to neuron interactions. Instead, it is ending with these neuron endings and then these swellings called varicosities, okay? And then we have our effector tissue. So this is our effector tissue, right? And this is our neuron, our varicosity, excuse me. This is our varicosity. So the first difference that we said is that this distance is greater. So I'll put 1 here, greater synaptic distance, excuse me. And the second difference is the presence of these varicosities. All right, let's draw the similar thing out on the other side for our parasympathetic system. Again, we've got our neuron, got our varicosities, and we've got our affected tissue. Our postganglionic neuron, our varicosity, okay? And remember, the varicosity is where we have the neurotransmitter stored. So on this side, I'm actually going to put here neurotransmitter stored within those varicosities. Let's at the bottom look at a closer view of what's really happening across this junction. So we've got our swelling or varicosity. And in this varicosity, because we're on the sympathetic side, what is our neurotransmitter? Who can tell me what our neurotransmitter would be on the sympathetic side? What would our neurotransmitter be on the sympathetic side? Any ideas? Norepinephrine, excellent, thank you. So on the sympathetic side, our varicosity is storing norepinephrine or epinephrine. Let me just make that clear here. It can be either one. Norepinephrine or epinephrine. Okay, so we've got these vesicles storing our neurotransmitter. Right, and I'm kind of going back and forth with the colors here, I apologize. We've got the same exact mechanisms of what we see on our regular synapse. So we've got our voltage gated calcium channels lining these varicosities when the action potential arrives. So the action potential is coming down the length of this neuron. When it arrives at the varicosity, it's going to trigger the voltage gated calcium channels. Calcium rushes in, right? We depolarize the membrane and we dump our neurotransmitter into the cleft, right? So those events stay the same as what we see with any synapse. They just have a little bit further to go in terms of that distance here. But then when they get over to the effector tissue, what receptors are they going to bind to? Who can tell me what receptors this specific neurotransmitter will bind to? And whether that would be fast or slow. Who can tell us what receptor this will bind to here?
Sodium channels or calcium? I think calcium.
Not ion channels. These are not ion channels, but we're thinking about sympathetic system here. We're thinking about norepinephrine. What does norepinephrine bind to? If anyone can remember that. Okay, so these are all called adrenergic receptors. So although we're kind of talking about this in a truncated fashion, make sure you're connecting this. So all the receptors that we just described, the mechanisms of, right, involving the second messenger, involving adenylyl cyclase, cyclic-AMP, et cetera, these are the receptors that we were referring to, okay? So these are all adrenergic receptors. Remember, adrenergic receptors are metabotropic receptors, in other words, slow receptors. Oops, metabotropic or slow receptors. That's why they were all coupled to that G protein that we described. And the examples here are all of the types. So like alpha 1, alpha 2, not alpha 3, excuse me, beta 1. So alpha 1, alpha 2, beta 1, beta 2, beta 3. Okay. And so while we're looking at this in a very general sense, we're thinking about norepinephrine binding to this receptor. Remember, these are on different tissues, right? Alpha 1 is in a different location, beta 1 is different, beta 2, beta 3, etc. So make sure you're making that connection, although we're kind of talking about these things differently. And so the cellular response will look different depending on where we are, also depending on what's being done to the second messenger. Am I exciting the second, am I activating the second messenger? Am I inhibiting the second messenger? Am I activating the tissue or am I inhibiting the tissue? But these will help to bring about the different responses based upon what we just spoke about. So from here, it gets a little bit more specific, but before that, the events are the same regardless of what tissue we're looking at, okay? And just to make the connection here even further, remember our affected tissue can be several places. Affected tissue can be smooth muscle, it can be cardiac muscle, it can be glandular tissue, it can be adipose. And so depending where we're looking at, we're going to have a different receptor helping to bring about this communication. All right, let's look at the parasympathetic side. So what is the neurotransmitter that we're going to encounter here on the parasympathetic side? So again, think about our varicosity here, which has X neurotransmitter. I'm going to wait for someone to help me with that. So what neurotransmitter are we looking at if we're thinking about the postganglionic neuron on the parasympathetic side? So this would be acetylcholine. Okay, remember, the acetylcholine is secreted by cholinergic neurons. Excellent, yes, thank you. So I kind of jumped the gun there, but someone is sharing this in the chat. Acetylcholine is what we encounter from the parasympathetic side, okay? And just to even go a step further in reviewing this, remember all the preganglionic neurons release acetylcholine, but on the postganglionic neurons, the sympathetic is norepinephrine, but the parasympathetic is also acetylcholine. So therefore, all parasympathetic neurons, both preganglionic and postganglionic, are cholinergic. They all secrete acetylcholine, okay? And so we've got our acetylcholine stored in our vesicles on this side. Okay, it's going to have the same event. So the events are kind of repetitive here. We've got depolarization happening when these voltage gated channels open up, calcium rushes in, we dump that neurotransmitter into the cleft, and it can cross and bind to a very specific receptor. So what type of receptor are we encountering here on our effector tissue? If anyone can remember this from what we said last time. Excellent. Yes, thank you. It's a muscarinic cholinergic, very specific. I'm really glad you were very specific with that. So it's not just the cholinergic, because we do have nicotinic cholinergic, which are not found at these junctions. Those are found at the neuron to neuron junctions or on the adrenal chromaffin cell. Here at the neuroeffector junction, if it's a cholinergic junction, it will be a muscarinic cholinergic receptor, which is also a metabotropic or slow receptor. So here we have our muscarinic, cholinergic receptor, which is also a metabotropic or slow receptor. Oops, metabotropic or slow. And although we did not speak about the mechanism of this exact response, we're not going to actually go into the details of this because it's not different from different cells. So we're not going to go into the details of the mechanism. It is a slow response. It's going to have different second messenger, and it's going to bring about the response on parasympathetic tissues, okay? And so that's how we bring about that parasympathetic neuroeffector signal here, Okay. Now we also want to remind ourselves that we have to clear the junction. But in terms of clearing the junction on the sympathetic side and clearing the junction on the parasympathetic side, it's going to be different. We have our amplifier, not our amplifier, excuse me, our degrading enzyme. So our degrading enzyme, which is acetylcholinesterase, is only present on the parasympathetic side because acetylcholinesterase can only degrade acetylcholine. So here, acetylcholinesterase is the name of our degrading enzyme here, right? ASC means it's going to cleave or cut or break down acetylcholine. And then that's how we can chop this up into smaller pieces. And then we also had two other routes of uptaking this. We can just diffuse it away from the junction, or we can actually reuptake, excuse me, these whole neurotransmitters, we uptake them back into this varicosity, and we package them to make a new vesicle, all right? So we can degrade this into smaller pieces and diffuse away. We can diffuse away as is, or we can reuptake it in its whole form as is. On this side, we don't have a mechanism to degrade norepinephrine, okay? But what we can do is reuptake norepinephrine in its full or whole state. All right, so we can have this similar reuptake molecule present here. So I'll just label this. Okay, really important to understand acetylcholinesterase is not present at this junction because it has no function. It cannot degrade any other neurotransmitter but acetylcholine. Okay, I'll pause here and see what questions we have on this, and then we'll take it a little bit further in the remaining minutes. Okay. All right, so this is just summarizing, again, just going over this for a bit of repetition here. We've got the events that are taking place, and we want to understand that the events are different depending on where we are in the autonomic nervous system, what division of that nervous system. So we've got action potential arriving at our varicosity, the opening up of voltage-gated calcium channels, calcium triggering the exocytosis of our vesicles, of our neurotransmitter within that vesicle, and then they can cross the junction, which is a little bit wider here, remember, and then they can bind to their respective receptors, and then they can bring about cellular response. On our parasympathetic side, the neurotransmitter is acetylcholine. The response will be brought about by a muscarinic cholinergic receptor. The degrading enzymes acetylcholinesterase, and so we can degrade that enzyme. We can reuptake it, so repackage it to make a new vesicle, or we can simply let it diffuse away. The sympathetic side, the neurotransmitter is norepinephrine or epinephrine, both are sort of alternative there. That dumps into the cleft after the action potential stimulates that calcium channel. And then that combined to a different receptor. So we're gonna kind of lump this together as adrenergic receptors, but remember these are the beta 1, beta 2, beta 3, et cetera, et cetera. We don't have a degrading enzyme here, so just make that note. There's nothing to degrade norepinephrine, but then we can reuptake it back into the vesicle in its whole form, and we can make a new vesicle with new neurotransmitter. Now, the schematic here is showing the mitochondrion within the cell. And this is monoamine oxidase, not something that we're going to speak about, but this is where norepinephrine is made. And so we're going to see that here, not see it here on the parasympathetic side, because this is how we actually make that norepinephrine via the mitochondria, which has the monoamine oxidase that makes that. So when we recycle that, we can make a new vesicle, or we can make new monoamine oxidase from the mitochondria. Again, we're not going to speak about the details of that, just to show you why the mitochondria is being shown.